Welcome to another explainer. Today, we're taking complex, unseen worlds and making them crystal clear. Let's dive right in and unpack the incredible, high-stakes battle plans of two highly destructive microscopic pathogens. We are looking at a literal war going on at the cellular level. We're going to examine the invader's playbook, specifically looking at how two very different bacteria, Brucella and E. coli, execute their devastating attacks on their hosts. It's a pretty wild question, right? How exactly do these literally microscopic organisms manage to completely take down a massive 1,000-pound farm animal? And not only that, but how do they cross over to infect us humans through everyday sources like raw cow's milk? Well, to truly understand how these bugs devastate massive livestock populations, we have to look closely at their completely different operational strategies. They don't just rely on brute force. They've actually evolved highly sophisticated tactics to completely outsmart the host's defenses. Here is our roadmap for tracking these rival villains today. First, the microscopic invaders. Second, Brucella, the stealth infiltrator. Third, we'll look at the toll of Brucellosis. Fourth, E. coli, the gut hijacker. And finally, its 3 tactics of attack. All right, Section 1, the microscopic invaders. So our first subject is Brucella. Sure, biologically speaking, it's a gram-negative coccobacillary organism, which is a mouthful, but functionally, it's basically a deep-cover operative that lives entirely in the shadows to establish chronic, deeply rooted infections. Unlike pathogens that attack quickly and loudly, Brucella plays the long game. It establishes a persistent presence in the body, often without showing immediate clinical signs, by doing something remarkable. It actually thrives inside the host's own cells. Which brings us to Section 2, Brucella, the stealth infiltrator. But let's actually map out this stealth infiltration step by step. It starts super simply, ingestion, but from there, the organism forces its way through the intestinal epithelial cells, specifically targeting those overlying the Peyer's patches. It uses endocytosis, which essentially tricks the gut barrier into swallowing it whole and passing it right through, like a microscopic Trojan horse. Once it's through, it rides the lymphatic system directly into the bloodstream, a state known as bacteremia, which allows it to localize in very specific tissues. Brucella has a particular, dangerous affinity for reproductive organs, the placenta, the fetus, and mammary glands. Now here is the absolutely crucial and honestly kind of mind-bending part. Brucella actively resists destruction in a brilliant way. See, when a normal macrophage swallows a bacteria, this thing called phagosome-lysosome fusion is supposed to digest and kill it. But strains like B. abortus, they block this fusion entirely. Brucella actively survives and aggressively multiplies inside the very immune cells meant to clear it. It literally turns the body's primary defense mechanism into a highly mobile safe house. Just look at the sheer adaptability going on here. Brucella militensis targets sheep and goats, giving humans Malta fever. B. abortus goes after cattle and horses. Then you have B. suis hitting pigs and B. canis targeting dogs. It's an entire family of pathogens, each with a highly specialized key to a different animal's immune system. The way it seamlessly pivots across the animal kingdom and right into human populations is truly staggering. Section 3. The toll of brucellosis. For agriculture and the farmers on the front lines, this is the stark, tragic bottom line: widespread reproductive failure. Abortion is the most serious clinical outcome of brucellosis across most domestic animals. Because the bacteria specifically targets the reproductive organs in the placenta, it causes devastating losses. For instance, cows exposed to Brucella after puberty often develop persistent infections, and the usual outcome is abortion, typically between the 7th and 8th months of gestation. In infected sows, this tragedy happens between the 2nd and 3rd months. Across all these different target species, the physical toll is just massive. At the tissue level, particularly in the lymphoreticular system, Brucella attracts macrophages and forms small granulomas. As these grow, they undergo caseous necrosis. Basically, that's a destructive, cheese-like death of the central tissue. We also see severe necrosis in the placental cotyledons, completely wiping out the life support system of the fetus. The aborted bovine fetuses characteristically suffer from bronchopneumonia. And interestingly enough, in horses, Brucella abortus targets the ligaments of the neck, leading to painful, debilitating conditions historically known as pole evil and fistulous withers. Moving on to Section 4. E. coli, the gut hijacker. Okay, we are trading the slow, stealthy, inside job infiltrator for a rapid, explosive surface attacker. Colibacillosis. It's fascinating because E. coli is a classic Jekyll and Hyde scenario. Normally E. coli is just a standard inhabitant, just hanging out in the digestive tracts of mammals and birds without causing any trouble at all. But under the right conditions, certain strains possess different surface antigens that completely transform them from a harmless neighbor into a highly dangerous hijacker, causing severe enteric disease and widespread diarrhea across numerous species, including us. To really grasp how it makes this transformation, we have to look at its arsenal. Think of these 4 surface antigens as a highly specialized tactical weapons loadout that equips E. coli for its assault. First, you have the O, or somatic antigen, which is an endotoxin liberated when the bacteria's cell bursts. Next is the K, capsular antigen. Think of that as surface armor that boosts the bacteria's virulence. Then there's the H, flagellar antigen, providing the crucial mobility that drives pathogenicity. And finally, the F, fimbrial or pilus antigen. These literally act like microscopic grappling hooks, allowing the bacteria to firmly and aggressively attach to host cells. Armed with those specialized weapons, how exactly does E. coli conquer the gut?
Section 5, 3 Tactics of Attack.
Armed with those weapons, the bacteria executes one of 3 totally distinct and frankly brutal pathogenic mechanisms. Notice the incredible contrast in attack strategies here. On one hand, you have enterotoxic colibacillosis. This strategy relies entirely on chemical warfare. It colonizes the surface of the gut cells, the enterocytes, and pumps out toxins that trigger massive, life-threatening fluid loss. On the other hand, you have enteroadherent colibacillosis. This version? It doesn't bother with toxins at all. Instead, it uses those fimbrial grappling hooks to firmly attach to the mucosal cell surface and causes direct physical destruction, essentially wiping out and effacing the microvilli of the gut. Let's zoom in on the enterotoxic tactic first, because it's incredibly common in young piglets, calves, and lambs during their vulnerable first week of life. When this strain strikes, it's a fast-paced, urgent biochemical hijacking. First, it colonizes the gut surface. Second, it produces specific toxins, either heat-labile or heat-stable. Third, and here's where the cellular tampering really happens, the LT toxin activates adolite cyclase, raising intracellular cyclic AMP, while the ST toxin activates guanylate cyclase. Finally, this biochemical override forces the host's own cells to betray them, rapidly pumping out vital fluids and resulting in severe, rapid-onset diarrhea. Finally, we have the third tactic, enteroinvasive E. coli. If the first 2 were chemical warfare and surface destruction, this one, it acts like a destructive breaching charge. Found in humans, pigs, and cattle, these organisms don't just sit on the surface, they actually penetrate the enterocytes and can even extend deep into the lymph nodes. Once inside, they release Shiga-like toxins. These toxins aggressively inhibit the host cell's protein synthesis, essentially shutting down the cell's engine entirely, which leads directly to necrosis. This isn't just fluid loss anymore. This is the aggressive, permanent destruction of the intestinal lining.
So we've seen 2 entirely different masters of infection today. Brucella, the ultimate stealth operative that hides within our very own immune cells to create chronic, devastating diseases. And E. coli, the opportunistic hijacker equipped with grappling hooks and chemical toxins to rapidly conquer the gut. Seeing these brilliant but deadly microscopic battle plans laid out should really make you wonder about the ongoing war happening right now at the cellular level. With pathogens constantly mastering both intracellular stealth and rapid surface assaults, how can medical science and agriculture stay one step ahead in this endless evolutionary arms race? It's a huge challenge for sure, but understanding their playbook, that is the absolutely critical first step.